Ça me fait plaisir de vous introduire Dax Bourcier, qui vient directement de Moncton pour nous présenter son projet. Et dans le fond, ce qui est un drôle de hasard, c'est quand on a reçu son abstract pour le congrès, on se disait « Oh, qu'est-ce que c'est ça, une titinopathie? » Alors, et là, on regarde ça, puis on se dit « OK, ben c'est bon. » Et quelques semaines plus tard, dans le fond, il y a un collègue de l'université à Sherbrooke, mais à Moncton, qui m'appelle et me dit « Écoute, il y a quelqu'un qui veut faire un projet là, en génétique. » Il dit « Je sais pas trop. » Mais il dit « Écoute, t'es la seule qui fait ça dans notre faculté, dans notre programme en sciences cliniques. » Fait qu'il dit « Écoute, on va faire un Skype avec. » Et là, je vois le nom, je fais comme « Hein, Dax Bourcier. » Ah ben, j'ai dit « Le monde est tout petit. » Fait que finalement, ça va être théoriquement, officiellement, mon nouvel étudiant. Donc, d'abord, il est étudiant en médecine. Il est en 2e année. Puis, dans le fond, ce qui est prévu, c'est qu'il va prendre une pause pour faire sa maîtrise en janvier 2019 avec moi et mon collègue de Moncton sur la taxie de Charlevoix-Saguenay. Mais aujourd'hui, il vient vraiment nous présenter, dans le fond, les résultats de son stage d'été de l'année passée avec le docteur Aimez Marrero. Et c'est, dans le fond, ça lui a permis, dans le fond, d'avoir des résultats de recherche qui sont sous publication. Je pense qu'ils sont en écriture présentement. Et, dans le fond, il va vous présenter ça aujourd'hui. Et peut-être que dans 2 ans, il va vous présenter quelque chose sur la taxie au prochain congrès.
Bon, bonjour tout le monde. Premièrement, j'aimerais m'excuser parce qu'au début, je pensais qu'il fallait que je présente en anglais. Donc, ouais. Donc, c'est ça. Donc, mais regarde, si vous me demandez des questions en français ou en anglais, il n'y a pas de problème. C'est juste que mes slides sont en anglais, donc je vais continuer en anglais. So, thank you very much for the introduction, and we'll get into it. So, I'll present to you today a new homozygous mutation in the Titin gene, which causes a pure skeletal muscle pathology without cardiomyopathy. So, I have no conflicts of interest. This is a continuation of a summer project that I had begun last summer. So the agenda for today, basically we'll start talking a little bit about Titin, a little bit about the spectrum of diseases that it has, and then I'll introduce my case report that we did last summer and continuing, and then we'll talk a little bit about some interesting points and for future directions for this project. So let's start off with a couple Titin fun facts. First of all, it's the biggest human protein. So you might not have known that. We talked a little bit earlier about a big dystrophin protein. Well, this one's even bigger. Titin, if we were to put it into a protein, it would be about almost 36,000 amino acids. So it's huge. And it's thought that everyone has a different polymorphism. Basically, the role of this protein is it spans the sarcomere, so it ties the Z disc, and it goes all the way to the M line. And it ties around myosin as well. So it's a muscle protein. And because of this role, it also underlines the Frank-Starling law in heart physiology. And overall, there's 364 exons, and there's at least 127 pathogenic variants, as well as over 10 different pathologies in this gene. So let's dive into it a little bit more. Here we have a muscle fiber with myofilaments. And then if we go a little closer, we see the sarcomere. And to the arrow is where the Titin protein is located. So as we see here, it really spans the Z disc to the M line. And again, it's in pink. So if we look at the gene now, this is how it's divided. So the Titin gene, because of its huge size, like I mentioned, is divided into 4 parts. So there's the Z disc coding part, the I band coding part, the A band coding part, and the M line. Now, if we look into these different parts a little bit more closely, in the Z disc, basically this part's role is an anchor. So it anchors the Titin protein to the Z disc. And then the I band is a molecular spring. So this one acts as the spring between the Z disc and the myosin and allows for elasticity of muscle. And within Titin, another thing I want to mention is that there's 6 classes of isoforms. So basically, depending on what type of muscle it is, there's different isoforms. And the I band is where most of the differences lie. And it's in terms of elasticity, which makes sense. Now if you look at the A band, this part plays a role as a stabilizer. So it wraps around myosin and allows for a tight grip. The M line is more in terms of modulating expression and as well as turnover because of all its protein interactions that it does. Now, if we look at the principal diseases associated with each of these parts, I'm not going to go through these, but basically it goes from top to bottom into more common to less common. And in red, we have cardiac muscle disease, so purely cardiac muscle. And then in blue, we have skeletal muscle disease. And basically, we see that in the Z disc, I band, and A band, it is mostly cardiac muscle disease. And then in the M line, we see more blue, so there's more skeletal muscle disease. Now, you might notice that there are blue in the other bands too, but the CH stands for compound heterozygotes. This means that there's another mutation somewhere in the gene that as well, and they're all in the M line, okay? So moving on, we see that there's about 75% of the diseases that are pure cardiac phenotype. There's about 7% that's a mixture between both skeletal muscle pathology and cardiac muscle pathology. And then there's about 27% that's pure skeletal pathology. So today we're talking about pure skeletal muscle pathology. Our case report is one of those. So the first skeletal muscle pathology that was identified was tibial muscular dystrophy to be on Titin. So this was described in 1991 by Aimez from a Finnish population. And then in 2001, it was found that the likely gene was probably Titin. And then in 2002, it was identified to be on the M line. And this is what we see at the bottom right there. The little lightning is where they found the mutation to be on. And then with the advent of next generation sequencing, what happened is this. So there's 6 new pure skeletal muscle pathologies that were described. I'm not going to name them all, but basically we see that it scatters across Titin. So there's different mutations of skeletal muscle pathology throughout Titin. And also another important aspect is that these mutations are either compound heterozygote, so most of them are compound heterozygote with a mutation in the M line, and others are heterozygotes. So another thing I wanted to talk about is that in multiple different muscular dystrophies as well as cardiac disease, it's a mutation somewhere in this big protein complex. We saw half of this diagram already earlier with the dystrophin. So Duchenne muscular dystrophy is up there. And then there's also limb-girdle muscular dystrophies. There's Emery-Dreifuss muscular dystrophies. And there's dilated cardiomyopathies. So they're all related into this one big or multiple protein complexes. So now we're going to talk about my case report. Basically, I'm going to take you through the clinical progression of our patient. So starting with 2013, so when he was 13 years old again, he was first known to the clinics for the excision of a left scapular osteochondroma. Now, a little bit later in that year, he noticed he was starting to get a little tired, muscular fatigue, general fatigue. And his father was diagnosed with hemochromatosis, so he thought maybe this could be it, and he decided to go for a screening. And he doesn't have hemochromatosis, but they found serum creatine kinase levels at 13,000. That's 65 times higher than the upper normal limit, which is extremely high. And so obviously that raised a flag. And but because of the slow progression of his symptoms, he was only referred to a neurology clinic in 2016, which is where he appeared at our clinic in Moncton. Now, at this time, he had a little bit of progression in his symptoms, so a little bit more arm and leg weakness at the end of the day. And so we decided to do a muscle biopsy to have a better look at what was going on. And then again, his CK levels were still in the 13,000. So these are the biopsy results. On the left, we see necrotic fiber, and on the right, there is also necrotic fiber with macrophage infiltration. This is abnormal, and also it was a quadriceps biopsy. This is electron microscopy of showing membrane interruption. This is non-specific, but it is common in muscular dystrophies, where the arrows are pointing. And then this is kind of the finding that elucidated the team a little bit. It's the absence of dysferlin. So in the top left, we see it's an immunohistochemical stain for dysferlin, which is a control sample there. And below is the one of our proband. So there's a loss of dysferlin. Now, what this signifies typically is a limb-girdle muscular dystrophy type 2B. And so this was it was a suggested limb-girdle muscular dystrophy at this point in time. Except if we go along our little clinical timeline here, the symptoms didn't really fit too well with the limb-girdle muscular dystrophies. There's something else in there. And so to confirm, we wanted to do genetic testing. And that's what was done. Also, the studies for cardiac implication, respiratory implication, as well as sleep studies were done and were all normal. So the genetic testing, though, gave us this. And it was a little bit to our surprise that we found 2 homozygous mutations in the Titin genes in the A band. So outside of the M line, where most of the skeletal muscle pathologies are usually. And so this is what we see here. On the left is the change is just one methyl group less amino acid change. So we don't consider this one to be pathogenic, but the one on the right is the one that is likely pathogenic, which we'll see in just a little bit. But we see the proband at the bottom, which is homozygous for the mutation, and we also see that both his parents are heterozygotes. So we did the genetic testing in his parents as well, and they were both carriers of both mutations in the A band. And the parents also are asymptomatic, and they're in their 40s, 50s now. So. And the last thing is that there's presence of consanguinity in the family. They're thought to be third cousins. Now, if we take a closer look at the localization of the mutations on the Titin gene, we see that it is within the A band. And just a reminder, the A band is the area, it's the stabilizer of the protein. It's where it wraps around myosin to get a good grip. And then the table is not too important, but basically what it's showing is that the likely pathogenic variant is highly conserved throughout species, and that the change in amino acid sequence could cause a conformational change in the protein, causing damageable impact. So again, if we keep going, there's more creatine kinase tests that were done throughout, and again, it's still very high. And the last time that the patient came to see us was in January 2018. And at this point in time, the patient's now, he was just turning 19. There's a clear progression of his symptoms. So he's having difficulty climbing multiple flights of stairs. And even he told me that he had new steel-toe boots at work. And at the end of the day, he wasn't able to lift his feet anymore because of the weight of his boots. So this is all new, and then the physical examination showed similar progression symptoms, or signs, perhaps. So this is the latest neurological examination. Again, he was 19 years old. So we'll start by saying that there's wasting of the medial gastrocnemius muscles, as well as the tricep muscle group implication. And in his legs, lower legs, there's dorsiflexor and toe extensor weakness. And there's also difficulty at heel walking. And this distribution of signs shows a humeroperineal distribution. Next up, we also have bilateral scapular girdle muscle atrophy, especially in the rhomboid muscles. And there's also a left lateral winging of the scapula. There's a positive Gowers sign. and then there's difficulty initiating gait. So even when he's standing up from chairs, there's clear slowness, and he even describes himself as being slower than before. And there's absence of contractures, hypotonia, myalgia, and fasciculation. And the rest was all normal. So these last couple one is more, it signifies more of a proximal loss, which is typical in limb-girdle muscular dystrophies. So now let's take a look at the distribution of the muscle groups. Basically, again, we mentioned that limb-girdle muscular dystrophy tends to be a proximal muscle implication, as seen on the left. And Emery-Dreifuss muscular dystrophy, which is another one part of this big membrane complex that I showed earlier, this tends to be more a humeroperoneal distribution of muscle implication. So in the calves, in the gastrocnemius muscles, as well as in the tricep muscle groups. So what we're thinking is maybe our case is sort of a mixture of both. This is on the front and the back. This is what we think is the muscle distribution in our patient in terms of the physical examination that we've done on him. So we see that there's definitely aspects of both of these muscular dystrophies present in the symptoms and the signs of our patient. Now, I want to compare, I'm not going to go through all this table, but basically I wanted to compare with different either limb-girdle muscular dystrophies as well as Emery-Dreifuss muscular dystrophy, and there's a link between them. So first of all, the limb-girdle muscular dystrophy 2A is a mutation in calpine 3. Hold that thought, we're going to come back to the next line and talk about that. Limb-girdle muscular type 2B, this is the one that typically shows a loss of dysferlin. So this is the initial suggestion of what our patient had. So this is there. And then limb-girdle muscular type 2J, that's a limb-girdle muscular dystrophy that's found on Titin. It's the only other one found in this gene, and it's in the M line. And then the Emery-Dreifuss muscular dystrophy, like phenotype without cardiomyopathy, is probably the clinical symptoms that match, or the clinical phenotype that resembles what our patient has the most. And again, this was found to be a mutation in the M line of Titin. So we see that the age of onset is pretty similar, except for maybe 2B and 2J. The CPK levels in our case are very high, comparable to perhaps LGMD2B. And then there's no cardiomyopathy or cardiac implication. There's no respiratory implication. And the affected muscle distribution, again, it varies, but it is most similar to the EDMD phenotype. Our patient has clear wing scapula. Again, this is present in the EDMD as well as the 2A, but none in the others. There are no contractures. This is kind of the only, well, one of the tipping points to maybe it's not an Emery-Dreifuss muscular dystrophy, because these have a triad, typically starting with contractures. And then at muscle biopsy findings, it's variable, even within different limb-girdle muscular dystrophies. And so the only aspect was the loss of dysferlin, which we looked at earlier, that could suggest an LGMD2B. But other than that, it varies. So, calpine 3. Now, this is a nice diagram showing Titin and some of the big important protein interaction it has with the protein complex that I showed earlier. Now, LGMD2A is a mutation in the calpine 3 gene, and so we see that there's the clinical phenotype of that, which I just shown. And also, the loss of membranous dysferlin, which we saw in our patient, can be due to loss of calpine 3, okay? And then interestingly, LGMD2J, as well as the Emery-Dreifuss-like phenotype, both show secondary loss of calpine 3. So there's something going on there. And maybe some of the clinical phenotype is associated with this loss of calpine 3. The only thing, or the only problem, is that it doesn't seem to interact with the region where a mutation was found. So A-band hotspot. This is also an interesting concept. The M line was considered to be the skeletal muscle disease hotspot for a while. And then hereditary myopathy with early respiratory failure, or HMERF, was found to have a hotspot in the fibronectin 3 119 exon in the A band. And this is pretty well described. And so there's a hotspot there for this disease. But then in our case, the mutation is in the fibronectin 361 domain, and there are two other purely skeletal muscle mutations that were found very close by. So in the same fibronectin as well as in the one previously to it. So perhaps there's a little hotspot for an implication of this part of the gene in skeletal muscle pathology. And the other interesting thing is that surrounding this little area, it's purely dilated cardiomyopathy. So it's purely cardiac disease. Now, in New Brunswick, so I'm from New Brunswick and I'm also Acadian, there are small population pockets. And so consanguinity is common enough. And there's lots of diseases, especially rare genetic diseases, that tend to occur. For example, I forgot an I there at Frederick's ataxia, but it's been described in the French Acadian population before. And also with the advance of whole exome sequencing, and especially its use in the clinic setting, we're finding a lot more mutations in our patients. And while this was being done, we actually found another patient who was heterozygote for the same pathogenic mutation in New Brunswick. And so we're thinking there's probably many other patients that carry this mutation. And so the identification is very important. So for future directions, clearly the familial tree I showed earlier was small. Co-segregation analysis is key. We need to extend the genetic tree. We need to be able to identify if there's other patients that have this phenotype or that are homozygous for the mutation. And we need to do functional studies as well. One protein of interest would be the calpine-3, which we've shown to be associated secondary loss, or it'd be interesting to see if it is effectively lowered in our patient's case. And then the future is gene therapy. We're hoping for CRISPR-Cas9 or other therapies to evolve and to be able to help patients in New Brunswick and elsewhere to be able to cure these rare neuromuscular diseases. So these are my main references, and I'll open the floor to questions. And fun fact, Titin is the longest word in the world. Its chemical name is the background there. It takes 3.5 hours to read. Fun cocktail tip.
Nice presentation. I noticed that your patient has 2-point mutation. Do you know other patient that has only one of those point mutation, and does that lead to muscular dystrophy?
So thanks for the question. I guess there's two parts to this answer. One is yes, the other patient that we found does only have the one that we think is pathogenic or likely pathogenic. She's heterozygote for it. So the second part is it depends if there's heterozygote or homozygote, because we have both parents of our patient to be carriers and asymptomatic. So to date, we don't have any other homozygote patients with either one of these mutations.
I just noticed that one of the point mutation is a J2A mutation. And I immediately thought, it's possible to change an A to a J.
Yeah.
So that's the future.
That is the future. And that's why we want to get the word out there that we do have multiple patients in New Brunswick that could benefit from these gene therapies.
Other questions?
Thank you.
Thank you.